Hey everyone, Professor Laura here. Well, Darwin and his theory of natural selection was very important. Now we have another important discovery to talk about. Gregor Mendel and the discovery of genetics. So now we're actually going to start talking about mechanisms of inheritance and what people thought was going on before Mendel came around. Then we'll talk about how Mendel discovered the basic principles of genetics and the laws he got from this discovery. But before we go farther, what do you already know about genetics? It might be helpful to pause this video for a moment to write down your answer. If you're not familiar, genetics is a really broad field. It refers to the study of heredity. So we're looking how characteristics vary in one generation and how they are passed from that generation to the next. We do a lot of parent-child studies because we're looking at what characteristics were passed from parents to their children. But let's first talk about mechanisms of inheritance, or how people thought hereditary traits were passed from parent to offspring. So one theory before Mendel came around was called preformationism. So this is the idea that in the sperm there's a tiny little homunculus all curled up, and then when it penetrates the egg, it develops into a human being. Spoiler alert, we don't think that's what's going on. Now we have a theory called epigenesis that most of development comes from the egg, though of course we now know that we have equal genetic material from the father and the mother. But how that genetic material comes together to form a new individual, that wasn't understood very well at all. One of the theories at Darwin's time, and this is something he believed himself, was called blending inheritance. Let's look at an example. In this population, we have a bunch of very dark cows. Then we happen to introduce a single white cow. The next generation, there are going to be a couple offspring that are lighter in color because of this single white cow we've introduced. A couple more generations down the road, and now we have a few more gradations in color because uh, with the introduction of a single white cow with a bunch of dark cows, we're going to have, a little bit of blending, but it's still going to be predominantly dark because we only added a little bit of this lighter color. However, that's not exactly what we see. To get a better example of what exactly going on, let's talk about Mendel himself and what he did. So here we're talking about Gregor Mendel. He lived in the 1800s and he was a contemporary of Darwin. Today we call him the father of genetics. And he did many, many experiments on the common garden pea. He was an Austrian monk, and in his monastery, he had a garden. And the pea was both easy to access and easy to grow. One of the sad things about Mendel is we actually didn't know about his work when he published it. He published in a rather obscure journal, and it was not rediscovered until 1900. So even though he was a contemporary of Darwin, how he affected the field of science happened a little bit later. One of the biggest sadnesses is we actually discovered a copy of the journal that Mendel published in Darwin's study. However, it was uncut. At the time, books were sold bound and you had to cut them open physically. It was a way to keep them in good condition. So this is proof that Darwin never read Mendel's work. It's one of the biggest what ifs in evolutionary biology. What would have happened to our field if Darwin had happened to read Mendel's findings? But we'll never know. So let's talk about what Mendel did. So he had so many different types of pea plants, and he noticed that there were a couple different traits that happened in exactly two forms. Flowers were either purple or white. Plants were either tall or short. Seeds were either yellow or round, and they could be round or wrinkled. The pods could be green or yellow. The pods could be full or puffy or constricted, and the flowers could be axial at where different branches branch off, or at the ends of branches. And it was interesting that he always saw one or the other and no intermediate traits. So let's look at an example, and we'll use our plant height as an example. Under the blending model, we would predict that if we had a tall plant and a short plant, all of the offspring would be of medium height, and for forever after, all of them would be of medium height. But we actually saw this. If we have a tall and a short plant and we cross them, we get all tall plants. And then if we take that hybrid generation and we fertilize them with each other, we get a ratio of 3 tall plants to 1 short plant. This is very different. So let's look a little bit farther at this example. Mendel figured out that what he thinks was going on was there were individual particles responsible for these different traits. Hypothetically, if both parents had 2, there is a 3/4 chance of having a tall plant and a 1/4 chance of having a short plant. So here, these genes, we're using big A for tall and little a for short. The big A is dominant because we see if we have, we're seeing that the tall trait can mask the short trait. And we call the short trait recessive. So here, this theoretical idea that Mendel came up with is called the genotype, the makeup or genes that you physically have, and then your phenotype is the physical traits that we see. And this led to the theory of particulate inheritance. Instead of it being blended, the exact mathematical mean between the both parental traits, now we have the inheritance of discrete characters via genes that are independently expressed. So this was the first major contribution of Mendel, that we do not have blending inheritance, rather we have particulate inheritance. We can still occasionally get something that appears to be blending inheritance, but that's because we have many, many, many different genes all contributing to a single trait. So let's talk more about our genotypes. There's a couple words we use to talk about different versions. We can have homozygous. Homo, again, means same. So if you are homozygous, you have the same gene at that location. If you are heterozygous, you have different versions at that location. One of the most important words we're going to talk about in genetics is the idea of a gene. The most basic idea, or most basic definition of this word, and that this is what Mendel himself came up with, is a molecular unit of heredity. Now that we know a little bit more about genetics, we call it a specific sequence in the genome that codes for a protein. However, this can get rather complicated. In this example, here we have hemoglobin here, and we have 4 different subunits. They all have their own sequence. So do we have 4 different genes or 1 gene with 4 parts? That's where it gets a little bit fuzzy. We also have many things in our genome that don't actually code for a protein, but still have a specific unit. So now we generally include functional RNA as a gene, and it doesn't necessarily have to code for a protein as well. So sometimes using gene and going back to the original definition of a molecular unit of heredity is even more useful. There's a couple other words that are helpful to know: genes, alleles, and loci. So we just talked about the gene. That is the trait we're talking about. The allele is the form. So here we have a red allele and a blue allele. And the locus is the physical location on the chromosome. So we already talked about the ideas of dominant and recessive, but there are actually a couple different systems of dominance that we see. So here we have some chickens. We have a white chicken, we have a black chicken, and then we have a black chicken with a little bit of white specks. This does not fit Mendel's classic idea of dominance. We call this incomplete dominance. So our speckled chicken here, it has 1 black allele and 1 white allele. But you can see that there is more black than white in this chicken. So black is a little bit dominant, but it's incompletely dominant in this situation. We also see different situations that we call codominance. In a codominant system, both alleles are expressed equally. This can be pretty rare, but one of the easiest examples is blood types. So a type A person has A proteins on the outside of their red blood cells. Type B has B proteins. If you are type AB, you simply have both. Type O, you actually have none of the above. Interestingly, it was originally written as the not symbol or nothing in math, and that eventually transitioned to being called type O. So we can see some examples in the plant kingdom here. There is a lot of incomplete dominance or codominance when we're talking about flower color. So here we have red and white being crossed and we end up with a pink flower. Not always, as you saw with our example in our pea plants, that those flowers do have classic Mendelian dominance, but in other instances we will see systems like this. So now that we've talked about what Mendel did, let's talk about what he discovered. So his first law is the law of segregation. Segregation is that two alleles of a gene found on each pair of a chromosomes segregate independently of one another into gametic cells. This means that meiosis happens. So each allele is separated and it's 50-50 whether you're going to get this allele or that allele from either parent. And it is completely independent. His next law is the law of independent assortment. This means that genes found on different chromosomes are sorted into sex cells independently of one another. This law means that each gene is independent of each other and that we shouldn't expect to see traits sorted with each other. We can notice that when we do things like this, when we look at two traits at a time. If there was dependent assortment or traits sorted with each other, we would only find a few possible combinations when we're looking at different types of traits. Instead, we see a wide variety of possible offspring when when we're tracking two traits at the same time. In this example, we're tracking both the color and shape of our seeds. And lastly, the easiest law to understand is the law of dominance. A dominant allele will always mask a recessive allele under this system. So we can go back to our example here. Remember, we have our genotype, or the genes that this individual has, the phenotype, what it looks like physically. We have our alleles. Big A is tall, little a is short. By convention, dominant traits are capital letters and recessive traits are lowercase letters. So Mendel's laws, again, we have our law of segregation, law of independent assortment, and law of dominance. These mean that alleles are independent of each other, chromosomes are independent of each other, and that some alleles mask others. What we talked about here is something called Mendelian genetics, and this just means genetics that follows Mendel's laws. These things are pretty simple and straightforward, though of course, there's a lot of genetics that doesn't follow these laws. We'll get to that later. So, what did we learn from Gregor Mendel? Can you explain it in your own words?